Good afternoon, Rotarians and guests. I know it's raining, but it's still a good afternoon. I'm Teresa Holmes, President of the Rotary Club of Madison. Welcome on our nice fall, rainy day, right? All right, well, thank you for joining us. Today's meeting is sponsored by Ma and Pa's Bluegrass Band. Jeff and Angela Bartell, our very own. And our thanks to them for their support of today's luncheon. With that, Angela will be leading us in the singing of our patriotic song, You're a Grand Old Flag, with Jeff Bartell on the piano, and then Jeff and Angela and the Bluegrass Band, including their sons, Nick and Chad, will lead us in the singing of our feature song, You Are My Sunshine. And following our music, our guests will be introduced by Aaron Embry.
You may be seated. All right, y'all. COVID isolation meant many things to many people. But to the Bartells, it meant a golden opportunity to take up new instruments and form a bluegrass band. I declared my long-held desire to play the fiddle. Son Nicholas said he always wanted to play the banjo. Son Chad already played the guitar, and he mumbled something about not having to practice much to play with the likes of us. Thank you, Chad. Jeff, it turned out, had always envied the electric basses laying down a bass line behind bluegrass music. So we purchased our instruments online, we found video lessons, and we began to practice about 6 months ago. Proficiency is the goal, but we're sure having fun, and like it or not, we're going to share with you our progress to date as we lead you in our song, You Are My Sunshine. Now notice, oh, you can't see it, this is a problem, but listen to these instructions. There will be a musical introduction. Then I will direct you in a chorus and then a verse. Then stop singing because there will be a musical interlude. Then I will lead you in a verse and a final chorus. At which time I expect you to explode with applause. All right, gents. My sunshine, my only sunshine. You make me happy when skies aren't gray. You'll never know, dear, how much I love you. Please don't take my sunshine away. The other night, dear, as I lay sleeping, I dreamed I held you in my arms. When I awoke, dear, I was mistaken, and I hung my head and cried. I'll always love you and make you happy if you would only say the same. But if you leave me to run another, you'll regret it all someday. Shine, my only sunshine, when you are happy, when skies are gray, you never know, dear, how much I love you. So So don't anybody think you're ever too old to do anything. One song only.
Good afternoon. Boy, that performance sure made me happy on this rainy day. I have the pleasure to introduce our guests today. If they could please stand and remain standing so we can recognize them as a group. Chad Bartell, guests of Jeff and Angela Bartell. Nick Bartell, guests of Jeff and Angela Bartell. Mike Beresford, guests of Teresa Holmes. Teresa Holmes. Thank you. Been practicing these names. Carol Ruley, guests of Jim Ruley. Ricardo Omarone, guests of Anthony Gray. And Brynolf Ernsten, guests of Martin Loth. Thank you for being here.
Thank you, Bartell family and Aaron. Listen, if the president can mess up a name, surely the guest introducer could mess up a name. All right, wonderful. All right, Maggie Porter Krantz is a member of our Community Projects Committee, and she has an announcement. Maggie?
Hello. Thank you, everyone who's already contributed to Soaptober. As I shared last week, our goal through the month of October is to raise toiletries, or to collect toiletries for Porch Light. The Community Service Project Committee is organizing it. As you know, last year we raised so many socks that they still have some. So this year, please join me in making sure that next year they still have enough soap that they say no. Also, the Community Service Project has a lot of other projects coming up, including this week, tomorrow, which is supposed to be lovely, collecting native seeds and helping distribute those. So look for more announcements on that. But lots of donations came in today. We also set up an Amazon wish list to make it even easier. So if you just want to go on your phone, we linked it on the Facebook, and it's also in the email. You can give that way as well. And those donations go right to the office. We're also going to have, for Soaptober, we're going to have a happy hour next Thursday from 3 to 5 over at Gymnast. So please join us for that. Thank you all.
Thank you, Maggie. Maggie's idea about the Amazon items for Soaptober is I just wanted to remind the members that when you're ordering for Amazon, you can use the Amazon Smile program and select our Madison Rotary Foundation to benefit from all of your Amazon purchases. You basically go into your Amazon settings, turn on the Amazon Smile option, and then you can enter Madison Rotary Foundation. Be sure to look for Madison Rotary Foundation in Madison because there are several Madison foundations listed there. Thank you again, Maggie. Our club board of directors met last Monday night, and I wanted to let you know that one of our members, Deb Archer, needed to resign from the board because she will be spending more time away from Madison, which won't allow her to participate regularly. Although we're sad to see Deb leave, the board actually has accepted her resignation, which is effective at the end of the month. In Deb's place, our board has named Scott Strong, who was last year's Sergeant at Arms, to replace Deb on the board with a term ending June of 2023. Normally what happens is the individual who has the most, or who would have had the most votes the previous election is actually proposed to the board, and Scott Strong was that individual. So Scott is the executive director of RISE Wisconsin and has been a member of our club since 2008. We thank Scott for agreeing to serve, and Deb Archer as well, who was also our member experience team leader. And our thanks also to Mark Clear, I believe, who is also in the room, who has agreed to fill in for the remainder of the Rotary year as meeting experience team leader. Thank you. Scott Strong is also co-chair of our club's Diversity, Equity, and Inclusion Committee, along with Ann Lucky, and he is actually going to come up and give us an update on the important work on this committee. Scott?
All right, thank you, President Teresa. I'm put to work right away. It doesn't take long to get at the podium, I guess. So Ann Lucky and I are honored to be the co-chairs of the Diversity, Equity, and Inclusion Committee this year. It's actually been in existence for quite some time, so there are a lot of co-chairs around the room and chairs that have kind of led the way to make it, prepare the two of us to kind of take over this year, so we're grateful to all of them. The other nice thing about this committee is it's actually one of the larger committees in the club and has really, really good engagement at each one of the meetings. So I'm really happy and proud and honored to serve alongside a lot of Rotarians who have DE&I at the top of their priority list for this club. So what I'd like to do is go through a little brief history of the DE&I committee. I'll raise this up a little bit. And there's this handout at most of the tables. I noticed that my table doesn't have this, but at most tables you have this handout. It's the history of kind of where we've come as a club. So in 2013 to 2016, we were called the Racial Equity and Inclusion Committee. That was started basically following the Race Equity Report. We noticed within Dane County we were not doing very well for our citizens of color in this community. And so that kind of prompted Rotary to take action and begin a committee to focus on that. And it was focused a lot on race, which was really important at the time and still is. Although the Diversity, Equity, and Inclusion Committee now we really are looking at all levels of diversity in the club, but we still need to make sure that we're focused on the racial equity as well. In 2017, the committee and some others participated with the YWCA and a diversity training, which kind of launched us into some deeper dive into the work that we needed to do as a club. We brought Maria White in, and many of you might remember her talking about inclusivity and what that means. So as we're slowly kind of building the educational component to our Rotary Club. In 2017-18, we added some questions to the Rotary Club Annual Satisfaction Survey, and we categorized those by age, gender, and race. And we ran some focus groups that year to get more information from our club members in terms of what's important and what we need to focus on. In 2019 through 21, we really started focusing on some traction to action. So essentially looking at education and community service, doing some benchmarking in our club. We shifted the satisfaction survey to an engagement survey, of which I think you probably all got this week in your mailboxes, so please fill that out. That's very helpful to us. We established some REI goals and objectives, and we set some targets for ourselves, mainly to have at least 20% of our programs focused on DE&I. And then presently, we are currently, we've renamed ourselves to DE&I to make it more inclusive and not focus just on race, but all areas that we need to be more inclusive and accepting of. The D&I committee now reports directly to the board. We have a co-chair model that started a couple years ago, which makes it a bit easier for us to stay on top of our goals and objectives. And we have, currently we are engaged in monthly meetings with our district governor and the Rotary Club of La Crosse and one up in the Eau Claire area talking about D&I. It's a topic for them as well. So that's kind of a quick history of what we have done so far. It's 8 years in the making, and I think we're doing really well, and it's going very strong. One more slide. And then currently, just want to give you a little bit of update. We have some goals set. One is to have at least 10% members of color by 2022. Currently, at this point, we're at 10.1%, so we're meeting our goal. Our members of color resignations are down, which is outstanding, and we have a goal to have at least 20% presentations on diversity, equity, inclusion, or have members of color presenting, and we're at 17% with programs on equity and diversity, which is outstanding. So we've come a long way. We've got a long way still to go, and we know along the way there's going to be some rough roads. There's going to be some challenges, and what we just ask of all Rotary members is to actually lean into those and educate yourself and learn. So with that, I'll turn it back to our president.
Thank you, Scott, for that report and the success of this group. You know, I will say this briefly. Many of you who don't or may not know your president is all about building bridges. And so several years ago, when I was a member of this committee, and our goal and effort when we were moving from race equity to diversity, equity, and inclusion was to reflect not about race and industry and, you know, all of the diversity specifics, but to open up the opportunity for this club to become more welcoming. That's where we placed our focus. We have done very well, and I mean, many of you in this room, if you are actually serving on the Diversity, Equity, and Inclusion Committee, could you please stand? Yes. And many of you who are not standing are also supportive of the work that we have been doing. And so my thanks goes to all of you because, again, it is more specific about our efforts to reflect what's happening in our club that's more welcoming in our community, and we are doing that well. So thank you. Thank you for your report as well, Scott. All right, our new organizational structure that was launched on July 1st is now showing on the screen. Not really that easy to see, but it's there. So, but we have reached the end of our first quarter, and with this new structure, we wanted to take some time to really give more reflection and awareness on our 5 team leaders who presented updated reports at our board meeting last Monday. So those 5 team leaders, if you're in the room, would you please stand? Mary Thompson, who leads our member experience team, Deb Archer, who is currently leading meeting experience, but Mark Clear, who's in the room, will be taking over as of November 1st. Please stand. As I mentioned earlier, Doug Dittman, who is our service team leader, Sandy Morales, who leads our youth service team, and Charles McLymans, who is our philanthropy team leader. These are our organization's new team leaders that reflect and add more continuity and synergy within our community engagement and our committee work. Thank you all for serving. We're getting some good feedback from our team leaders, and they are now holding regular meetings with their committees and chairs within their groups so that they're able to share ideas, and their pictures are on the screen, as you can see. And we are very thankful that they're providing very good suggestions to committee chairs, and we'll keep you updated on the progress of this new team and its approach. All right, announcements. All members, as I think Scott mentioned this earlier, received a link to our annual member engagement survey last Thursday. And so I'd like to remind everyone to please take the time to complete that survey. Your feedback is very important and will help club leaders evaluate what you, members, feel is important for our meetings and our activities as we connect, grow, and serve together. The deadline for the survey is October 31st. Our club is also participating in the Rotary District Ride October Environmental Days by teaming up with the Clean Lakes Yahara Project to collect seeds at Yahara Heights, the county park. So tomorrow we are collecting seeds from 2 to 4 p.m. So if you're able to join us, and we have a sign-up sheet at the button box table. So hope you'll be able to consider attending that event. I'd also like to thank Larry Hands and Karen Kendrick Hands, I think I saw who were in the room, for hosting our Going Green Fellowship kickoff or kickstart event last Saturday at their home. We had a great turnout for this event, which included some explanation of this complex decision-making process that went into selecting appliances, materials for internal and exterior working of the home, mechanical systems, so that no fossil fuels were burned in the operation of their new house. It was really exciting. We also had some great conversation around the dinner table, and the weather was great. So I posted some photos of that event on our club's private Facebook page. So you can go to that site to check that out. Renette Maurer has also, just a reminder, has donated 2 tickets to the UW Badger game versus Army this coming Saturday, October 16th. So they're suite tickets to the Hilton Suite, and for a donation to our Madison Rotary Foundation, those tickets can be yours. Just letting you know, the president has placed a bid on those tickets. Doesn't mean you can't bid. So if you're interested, please come down to the podium following the meeting, and outbid the president. Thank you. All right, the best time outside of the program, right? Birthdays. We have birthdays to celebrate with a bit of humor and wisdom that complements our Rotary mission. We also encourage our members to make an age-appropriate gift to the Madison Rotary Foundation, rounded up to $100 for our club's Synergy Scholarship Fund. All right, Rotarians, if you are in the room when I call your name, please stand. October 10th, Ralph Hawley. October 10th, Britt Peckham. October 10th, Jane Wagenke. October 11th, new member Jeff Gerritsen. October 11th, Willie Larkin. October 13th, new member Kyle Geisler. October 14th, Bob Block. October 14th, Jerry Thane, who shares a quote from Eleanor Roosevelt: "We would worry less about what others think of us if we realized how seldom they do." I like that. October 16th, Laura Gallagher. Thank you to our celebrants and their contributions to the Madison Rotary Foundation. Rotarians, please join me in wishing them a happy birthday. We have a few members in the news this week, but before I get to those, I wanted to let our members know that Scott Hammerson's mother, Carolyn Hammerson, passed away on October 6th in Mount Horeb. So we extend our condolences to Scott and his family. Other members in the news, Maria Averell Stroud was pictured and quoted in an article titled "A Filipino View of American History," the Cap City Hughes, October 4th. Congratulations to Roberta Gassman, who I think I saw in the room as well, who received a Leadership Impact Award on October 2nd from the Hmong Institute at its 45th year of the Hmong Diaspora to Wisconsin Banquet, Wisconsin Politics, September 28th. And then Oscar Morales, who was pictured and quoted in a cover story article titled Sitting at the table, Oscar was telling or has talks about networking and helping the community set the agenda. Capital City Hughes, October 4th. Now, on to our program. We hold our monthly board meeting generally at the first Monday of the month. And so we held our October meeting this past Monday. And so I encourage members to review the board minutes that we post in the members login section tab of our club's website. We want members to see our current board members, so we ask one board member to introduce a speaker each month. This month, Charles McLymans will make the introduction. Charles has been a member of our Rotary Club since 2017 and was previously a Rotarian in Naperville, Illinois, and also in Milwaukee. Charles is Chief Executive Officer of Holy Wisdom Monastery and is in his first of a 2-year term on our board. I will now turn the podium over to Charles, who will introduce our speaker.
Thank you, President Teresa. Our speaker today is Dr. Jon Young, who has titled his presentation, Making of a Drug from Bench to Bedside. Dr. Young received a BS degree from the University of Minnesota's Duluth campus. He earned a PhD degree from Michigan State University, and he completed postdoctoral work at the UW-Madison. Dr. Young spent 20 years with Merck before joining Celgene, where he built and led the high-throughput chemistry, purification, and analytical chemistry departments. In 2018, he came to Madison to build and lead a new academic biotech at the Wisconsin Alumni Research Foundation. Dr. Young says that for the past 30 years, he has had the distinct privilege of performing research to better understand the biology of disease and to develop therapeutic drugs to improve the outcomes in patients that are suffering from debilitating diseases like cancer, rheumatoid arthritis, diabetes, and mental health disorders. Today, he's going to share the process of making a drug, the length of time it takes, and the expenses involved in the research. We look forward to your presentation, Dr. Young, and we have made a contribution to the Rotary International Polio Plus Fund as a way to say thank you for speaking with us today. I also want to mention that we will have a Q&A afterwards as time allows, and that the microphone will come to you. Let's welcome Dr. Young to our podium.
Thank you, Charles, for that kind introduction, and Regina and Teresa for the invitation. My favorite tie also thanks you, because it was wondering if it would ever get out of my home office again and out into public. So it's really great to be here. And Jane, thank you very much for all your help with the logistics, getting this presentation put together. And so, let's see. Terrific. So good afternoon, everybody. I've had the distinct privilege to be able to work for the past 30 years in the pharmaceutical research and development field. I've been working side by side with some of the most creative, insightful scientists that the world has to offer. They come from all types of different disciplines, like from chemistry and biology, biochemistry, pharmacology and toxicology, as well as clinicians as well to move the compounds into clinical trials. The one thing that we all share in common, although our backgrounds are different and unique, and we all have an important role to play in the drug discovery process, is that we're all committed to the singular goal of trying to bring forward new medicines to treat or cure some of the most debilitating diseases with unmet medical needs that the world has to offer. I've spent most of my career in the past 15 years in the fields of cancer and autoimmune disease. And before that, I did a lot of work on CNS disorders such as Alzheimer's as well as depression. So I've learned a tremendous amount in these last 30 years about biology and the biology of diseases, but also on the overall processes to put a drug together. And so from talking with all Also involved in that, of course, is you need to come up with the right biological target. You then have to optimize the drug. You have to do studies on it to make sure that it's safe and efficacious. You also have to patent the compound and work with the patent office. You then need to move that compound into clinical trials and work with the regulatory agencies such as the FDA. And so in talking with family and friends over the last number of years, I realized that these processes, the timelines, the constant failure that we get used to in our business, 97% of everything that we do in research and development fails before it gets to become a product. And we get used to that type of failure, but it's not really failure unless you don't learn from it and you can grow from the experience. And so, today I hope to sort of share with you some of those trials and tribulations, demystify the process, somewhat. So I hope that we'll be successful in doing that. And so today's talk is broken down into the 3 very brief sections. We'll talk a little bit about the end-to-end process, understanding how normal biology becomes sort of a diseased state, and of course, the designing of a drug. And so as I was thinking about this, I was trying to figure out, how do you talk to a broad group of audience that may or may not be scientifically trained to think about the drug discovery process. And as I was sitting here watching the Olympics this year, I kind of came upon this particular metaphor. I'm an amateur runner. I've been running all of my life. I understand what it takes to train. I've had a goal of hitting a certain marathon time for a number of years. I've never made it, despite 25 years of trying to make it. But that's kind of the point. Everybody can work at it, but not everybody is going to be successful. And so as I was thinking about this and thinking about women's gymnastics, for example, what does it take to become a gold medal gymnast? And so doing a little bit of research, at any moment in time, there are 70,000 women that are part of USA Gymnastics. And they begin training at around age 5 on average. Of those 70,000, only 1,500 of them will ever be skilled enough to pass the requirements to compete at a level 10 to compete in college. And of those, only 50 of those will become good enough to be considered elite. And so that's 8% of these elite athletes. Only 4 of them are going to make the all-round team for the Olympics, which is about 8%. And of course, this year there were 12 different teams competing for the overall team gold. And so 8% times 8% turns out to be around less than 1% of the elite women will ever win a team gold medal. And so it gets even worse if you think about how many people actually started in USA Gymnastics. So in all, about 11 to 14 years of training it took for them to get to the elite level and become a gold medal. Luckily, the odds are slightly better for making a drug, but not much better. And so, I think as we're sitting around and being bombarded on TV or on social media by pharmaceutical direct-to-consumer advertising, we start to hear some phrases from time to time. For example, the AbbVie group down in Chicago, when they talk about Humira, they'll say Humira works by blocking TNF receptors. Pfizer will talk about a drug called Xeljan, which blocks JAK enzymes. And maybe Merck will talk about Keytruda, interacting with an enzyme called PD-1. And so what do they all have in common? Well, all of these are biological molecules. TNF, JAK, and PD-1 all belong to a type of biological molecule called a protein. And if you think back to your biology class that you took, all of your genetic information is maintained in your nucleus, in double-stranded DNA, and the DNA codes for all of the proteins in your body. It first has to be converted to mRNA. Of course, we're hearing all about mRNA with BioNTech, Pfizer, and Moderna these days. and it's the mRNA that gets transcribed to become a protein. So a biological molecule can be DNA, mRNA, or protein. A target is thought, sort of described as a biological molecule that is a driver of disease. A therapeutic hypothesis would be, let's design a molecule like Humira to bind to and block TNF-alpha, and that will block the overproduction of inflammatory signaling cascades that lead to inflammation. And of course, the drug then is the therapeutic agent like Humira that does that. So we're not going to get real technical. That's about as technical as we're going to get here. But it's just a few terms and definitions that I think are sort of helpful as we think about this. And so, much like training for USA Gymnastics, drug discovery is broken down into a number of discrete steps. The first thing that has to happen is you have to identify a biological target and correlate it and validate it for a particular disease. And so when AbbVie was thinking about Humira, the first thing that they did was try to understand, well, what is causing the rheumatoid arthritis? What is it that is driving that particular condition? And they understood through trial and error and a lot of great research that it was a receptor called TNF-alpha, as it turns out, that was responsible. That was a protein that was locked in the on switch. So TNF is actually a natural ligand. We don't have to get into the science, but it's kind of like the accelerator for the immune system. And if it's stuck in the on position, it's producing far too much of that particular protein, and it causes inflammation. Every cause and effect has a braking mechanism, and there are brakes on the process that should stop that overproduction. But anyway, so it takes about 2 to 10 years to explore a target, validate it for a particular disease, and it's at that point you might start a preclinical drug discovery effort. So most of the basic research happens through NIH and NSF funding and happens in academia. Pharmaceutical companies get involved at this stage where it's now a known target, and we need to go through about 4 stages of preclinical drug discovery to discover, develop, and optimize the drug itself. There are dozens of criteria that need to be met. About 67% of the things that we're working on here are going to fail. They're going to stop, and it takes about 4 to 8 years to get through preclinical drug discovery. If successful, it then moves on to 3 different phases of clinical trials. First, it's phase 1, where we see if it's safe. Phase 2, we try to select the dose that actually works and modifies the disease. And phase 3 then is where we do a large clinical trial to demonstrate that not only does it work in a broad population, but it's also, no new safety signals have been found. And so this usually takes about 5 to 8 years to complete, depending on the type of disease. Things like, cardiovascular disease, like hypertension, you can go through those pretty quickly, but something like Alzheimer's, Merck put a compound in, and Lilly did as well, and it took about 30,000 patients and about 8 or 9 years of phase 3 clinical trials, because the endpoint is cognition. It takes a long time to try to see whether or not the compound is working. And once again, 90% of all of these drugs in the clinical trials is going to fail. And once it completes phase 3, then it has to go through regulatory review. That takes anywhere from 6 to 12 months, and if you're lucky, the FDA will then they'll be looking to see if the benefits outweigh the risk. They will then write the label to how doctors can prescribe the drug. And so all in all, about 8 to 15 years is required from start to finish, and 97% of the drugs are going to fail. And for each drug that gets approved, the total cost is around $2 to $3 billion to get that approved. What I haven't said is, when do you file your patent? Typically, you're going to do it somewhere in, preclinical drug discovery. If anybody knows anything about patent law, which there's no reason you should, once you file a patent, you have 20 years from the time you filed it, if it's approved, to market that drug exclusively, or that widget exclusively. But we filed that patent, 2 to 3 years into preclinical drug discovery. We had 5 to 8 years of clinical trials. There's only about 8 to 9 years of patent life left to try to recoup your $2 to $3 billion investment. That's the nuts and bolts of the overall process. And so, as I mentioned, you have sort of TNF. Let's talk a little bit about disease. Okay, like I said, for every accelerator, there's a braking mechanism to maintain homeostasis in your body. When everything is in balance, the body is healthy, the body is functioning. And it's really about when things become imbalanced, when disease happens. For example, if you look at this teeter-totter here, and just sort of look at immune cells, here is too many immune cells, here is too few immune cells. When it's balanced, everything is functioning properly. But as we all know, immune cells have a lot of different functions. There's dozens of different types of immune cells. Some fight bacteria, some viruses. Others are doing tumor surveillance and trying to find tumor cells, surround them, and kill them. But what happens when you produce too many immune cells? This is where Humira comes in. So in some conditions where your body is overproducing too many immune cells, the body then starts, those immune cells start attacking itself. And this leads to, inflammation of the joints, and eventually, if left untreated, destruction of those joints. And so one therapeutic hypothesis would be, let's try to design a drug that can target those immune cells, put the brakes on, that overproduction. So that would be sort of a therapeutic hypothesis. And so let's take a look at this in a little bit more detail. And so biology is very complex. There is all kinds of signals that are coming from outside the cell to drive things happening inside the cells. There's an elaborate communication system. So if you think about, back in kindergarten, when the teacher went to the person in the front row and whispered something in their ear, and that person then would turn around and whisper the same thing in that person's ear. 28 people later, you hope the message is exactly the way it was communicated. But life isn't like that. Things go awry. That message gets disrupted. And so the same is true in biology. So here you have a protein that's on the outside of the cell. It wants to signal something to the inside of the cell. And so it's going to bind to a particular receptor. In this case, it's an inflammatory receptor. And so when that protein binds, it turns on the pathway. Okay, this pathway now is activated, and it then causes, chemical reactions to happen on the target, as signified by this letter P. You don't have to understand what that is. It's just a reflection of the pathway being turned on. But it also provides a place for other proteins to come and bind to it. That P can now whisper in the ear of this new protein called protein 2, and this signal then keeps going on and on and on. A different protein 3, this enzyme, then phosphorylates protein 2, and if this is not turned on... Oh, I'm sorry. Thank you. If that protein, is not turned off, that then leads to, inflammation. The same thing can happen if you have too few immune cells. As I mentioned, there are some immune cells that are designed to seek and destroy cancer cells. And, okay, there we go. So what happens if you have too few? My clicker's not working. There we go. If you have too few of them, this can then lead to, cancer. And so this is where, a drug like, Keytruda comes along to try to turn on your immune system. It binds to PD-1 that then releases sort of the brakes and allows more immune cells to be produced, and then you can go on to, kill the cancer, cells. And so finally, how do you go ahead and discover a drug? And so, we use the scientific method, sort of defined as a process of observation, hypothesis generation, and a lot of experimentation. And so the whole process of designing a drug is we need to figure out how to bind to a protein and block it. We have to have, chemistry involved. We have to have biology involved. We have to have pharmacology involved. And so as we're thinking about this, we might say, I want to block, inflammation. I'm going to hypothesize that if I design a drug that interacts with target X, it'll block inflammation. We then design a drug with organic chemistry. We put it through a series of tests in inflammatory assays and basically ask the question, did it work? If yes, is it good enough to be a drug? If yes, go on to clinical trials. It never happens that fast. As I said, it takes about 8 years to get there. And so typically the answer is no. And so then you go back and repeat steps A and B. And this is an iterative process. It usually takes 200 times around this iterative process to go from an early stage drug candidate to an actual drug candidate. And so what does this look like? So here's that same inflammatory target you saw earlier. You might remember that this particular protein, when binds to here, it can then turn the pathway on. We want to design a drug that binds to it and blocks the ability of that protein to bind to it. And so if we're successful, okay, we'll learn how to, okay. I tried to hit the reverse arrow, but it didn't go that direction. But essentially, if successful, if we can design a drug to bind to that and block that protein from activating the pathway, that could then turn into a successful drug candidate. Like I said, that takes about 5 to 8 years of experimentation, to get there. And so, in summary, discovery and development requires a lot of different collaborative partners, academia, pharmaceutical scientists, clinicians, regulatory agencies, statisticians. The timelines are highly protracted. The attrition rates are large. 97% of the drugs fail. The R&D investment, when you factor in the cost of 97% failure, is $2 to $3 billion per drug approval. And so, all of that said, with all of that failure, I've never had trouble getting up in the morning, knowing that you have an opportunity to go out there and help patients and live longer, healthier lives is really, really rewarding. So I'd be happy to take any questions you have.
Hello, thank you for your informative talk. I understand that sometimes drug development relies on stem cell lines, and that some of those come from sources that trouble people who are worried about whether it was the product of an abortion. How does that affect your development work?
That is a really good question. And of course, you don't have to look much further than University of Wisconsin-Madison to see where some of that research has been done. One of the leading stem cell researchers is Jamie Thompson from the University of Wisconsin, and he's now at the Morgridge Institute. Now, most of what I work on, small molecule drug development, biological drug development, we don't have to utilize those materials. That said, we're learning an awful lot about tissue regeneration by studying those particular types of materials. That's going to be very helpful, I think, in neurodegeneration, discoveries going forward. But by and large, I would say, it hasn't really directly impacted a lot of big pharmaceutical development at this stage. But it's an early field. I mean, it's in its infancy at about 20 years or 25 years old. And so I think a lot of the lessons that we're going to learn from that research is in front of us, not behind us.
Thanks for your talk. I'm curious if you could speak a little bit about the nature of public-private partnerships in drug development. You've spoken about this long timeline, the failure, and the cost of that. Just wondering about how that is working well in some cases, or challenges in that nature of government funding versus pharmaceutical private funding for drug development.
That is a fantastic question. I mean, if you think about it, academicians, they're motivated by getting grants, by publishing, by giving talks. Pharmaceutical companies are, in it for the medicine, but it's also a for-profit entity. And so we're in it, we think about obfuscation. We think about trying to protect the intellectual property as long as we can so as to not invite in, competition. So WARF Therapeutics is trying to do exactly what you just described. We're trying to, teeter-totter on that swing set, if you will. I am building an academic biotech using WARF funding, but using biological discoveries on campus and trying to work together to come up with a therapeutic. It's hard because a lot of them want to take that new molecule as soon as we discover it and write papers on it. But we can't do that because we haven't filed patents. And so it is a really delicate balance. And we have to really try to, and I urge everybody that's coming, proposing things to us to take it into account that if this becomes a drug, if you want to share in the revenue of that drug, we're going to have to protect that structure. And so if we license it to a big pharmaceutical company for clinical studies because we can't afford it at WARF to spend $2 billion on the clinical trials, we're going to need help. That new company that we license the asset to, they're going to want to control the release of that information. So it's really trying to find a balance and find the right partners that understand that and are comfortable with it. And so the people, the PIs and professors on campus that work with WARF Therapeutics, by and large, are driven by curing cancer than they are about publications. And so it's that mindset, I think, that is the best candidate to sort of collaborate with us. And a similar thing can happen with all other types of research on campus. A lot of people are talking about Moderna utilized NIH funding in the development of mRNA vaccines. What people may not understand is people have thought mRNA was going to be a great strategy for decades. They knew it was the direct link between making a protein that is functional and you might be in short supply of it. The problem is we didn't know how to actually deliver that to the cell. It's not stable in human blood, and it can't get into a cell. And so how do you get over that? It took 25 years of research by Moderna and others to come up with nanoparticle formulations to actually solve the problem. It turns out that they solved that problem at just the right time, right at the time of the pandemic. And so the nanoparticle formulations teamed up perfectly with the knowledge of how to make mRNA vaccines, and that is our way out of COVID. So it's that said, Moderna, despite the government funding in the early process, they still spent billions of dollars on the clinical trials. They got some help from the federal government. Pfizer did not, and so it's a delicate balance for sure. It's not simple.
Long question, short answer, I hope. In the last 100 years since the Spanish flu, we probably only had two major events. One was AIDS and one was polio. When this came along and we came up with what we thought were some solutions, it took FDA a really long time. I mean, we kept saying, well, it'll be another 2 weeks, it'll be another week, it'll be another 2 weeks. And it seems like there were holdups. Weren't they looking at the data before it was presented so that they could accelerate?
That's not how it works. In fact, when you hear that a company has submitted their application to the FDA for review, that's when they get it. They're not looking at the early data. That data is guarded, and only a safety data monitoring board that's independent of the company and independent of the FDA can see that data, and they can only see the data at pre-specified timelines. And so I think Pfizer just submitted their application for ages 5 to 11 last week, and they're going to review that data for the next 3 weeks and then vote on it on October 26th. So it actually is happening pretty darn fast, to be honest with you. If it wasn't COVID, if it wasn't a pandemic, it would take 6 to 8 months to review all of that data. They're putting it to the front of the line, as it turns out. So, I saw, an ex-FDA person, give a quote the other day. It's like, you know what, we're not going to, spend too little time. We're not going to spend, too much time. We're going to spend exactly the right amount of time to review and weigh the safety and the benefit. And thank goodness that they do, because it's a matter of life and death if they make the wrong decision. We can think back to some of the molecules like phthalimid that came out in the 50s that might have, not have been studied as thoroughly. And once it got into thousands of women of childbearing years, we know what happened with that particular molecule. So, they've got a tough job to do for sure. And of course, they're doing their best to keep the ideology and the politics out of the process and stay as independent as possible.
Thank you, Dr. Young, for speaking to us today. Rotarians, we are adjourned.